Hello everyone, I'm Akira Kakugo from Hokkaido University, Japan. Here I'd like to introduce my recently published work entitled Cooperative Cargo Transportation by a Swarm or Molecular Machine. This work was published in Science Robotics. In nature, we can observe thousands of examples of the collective behavior. Living in an organized group is a strategy to survive in a harsh environment. From school to cell, collective behavior is everywhere. This collective behavior is usually known as swarm, a species that may be weak on its own, but it becomes powerful as a result of forming a swarm. This is because swarm provide multiple advantages over the individuals like parallelism, flexibility, robustness, etc. Scientists have been trying to reconstruct such behavior in artificial systems due to the interest in these advantages. The functions that emerge from swarming are fascinating. A group of ants can transport several times larger food efficiently than the size of the single ant. Schooling of fish helps them to survive from the predators. Grouping also provides efficient locomotion and effective information transfer. It also helps to overcome obstacles to reach their goals. Another function of grouping is a self-healing ability. Mechanical robot systems working as a group already have been demonstrated to obtain the advantages of swarming. Here are some examples of the different sized mechanical robots. As shown here, a group of swarm robots successfully transferred information and transport heavy cargo. 1,000 centimeter sized key robot can be controlled to form different shapes. In addition to that, swarm of the micro-scale robot have been employed to perform various complex tasks such as construction of different complicated structures, sequential task execution, removing obstacles, solving mazes, etc. Despite such advancement in macro-scale robot, the development of a large number of small-sized robots is still challenging, which is crucial to enhance the scalability of the swarm robot. How can we fabricate a tiny robot? First of all, what is a robot? Robots are systems composed of actuators, processors, and sensors. To develop a large number of small-sized robots, we integrated photochromic azobenzene molecules as sensors, DNA-based molecular computers as processors, and biological motors as actuators to construct our swarm robot. Our swarm robots are very tiny and not possible to observe with naked eyes. They are 20 times smaller than the thickness of the human hair. A combination of DNA nanotechnology, bioengineering, and chemistry allow us to have a wonderful molecular swarm system. The swarming of the robot is regulated by employing light-controlled interaction between photoresponsive DNA attached to the microtubules on kinesin motor protein. In this molecular robot, microtubules were modified with azobenzene incorporated the single-strand DNA by azide alkyne cycle addition reaction. Azobenzene incorporated into the DNA strand can regulate their duplex formation by changing their melting temperature through cis-trans isomerization. Swarming of this robot was initiated by DNA duplex formation, which was induced by the trans-cis isomerization of the azobenzene unit in the single-strand DNA and the visible light irradiation. And UV light irradiation induced the dissociation of the swarming of this robot. This will be demonstrate the group formation of the molecular robot comprised of DNA and processors. In the visible light, we can see that swarm grows with time due to the structural change of the photosensors. The length of swarm can be more than 100 micrometers. With UV light radiation, dissociation of the swarm started. By varying the extent of the DNA interaction between the actuators, we can control the swarming. Now, one may ask, is it possible to utilize this molecular swarm robot for any task achievement? Yes, that is what we did. We made them work as a molecular transporter. To work as a transporter, three main functions are required: such as pick up cargoes, transportation, and deliver to the designated destination. Nature already attains such highly sophisticated process by a group, for example, and utilize this strategy to carry large-sized food. In our work, different-sized microplastic was used as cargo. To recognize cargo, DNA tag was tethered to the cargo. Let's see how a transporter can pick up and transport the cargoes. In this scheme, cargoes are shown as a green sphere, and single transporters are carrying cargoes. Another front, group of transporter can carry the multiple cargo at a time. What's the advantage of such cooperative transportation by a group of transporters? You can find an answer from the trajectory of the cargoes. Group of transporters are carrying cargo for a long distance. Transport efficiency was calculated considering the transported distance of the cargo by the transporters. It was found a group of transporters is able to transport cargo more than 5 times higher than that of the single transporters. Besides efficiency transportation, we also found that there is a significant difference in the size of the transportable cargo. Cargo size varied from 1.1 to 30 micrometers. Moving on the top show the cargo transportation by a single transporter, and at the bottom by the group of transporters. Single transporter could transport cargo up to 3 micrometers in size, but in case of the group, cargo size as large as 30 micrometers could be transported, which is a tenfold increase in size compared to the single transporters. Thus, pick up and transport cargoes were efficiently achieved by the group of transporters. Then our next step was to deliver the cargo to a designated destination. Here I am showing the idea of delivering cargo at a specific location with this scheme. A transporter can form group under visible light irradiation, whereas UV light irradiation can trigger the dissociation of the groups into the individuals. The dissociation of the group or the molecular transporter carrying the cargo may allow to unload the cargo from the transporters. The delivery destination was specified with the UV light irradiation at the predetermined position. In this movie, a group of transporters are observed to deliver a cargo at the designated location. The area surrounded by a purple line is a place in which UV light was irradiated. The density of the cargo at the location was found to increase with time. Thus, we could successfully demonstrate efficient cargo transportation by a group of the molecular transporters. So our findings are, first, group transporters are able to transport large and numerous cargo with more than 5 times higher transport efficiency. Second, the swarm robot can deliver the cargo at the designated destination. Here are the perspectives. This system is composed of approximately 5 million transporters with size of 5 micrometers. This tiny swarm system could have a wide variety of applications that cannot be achieved by the mechanical machine constructed through top-down approaches. We expect our swarm robot will be helpful for intensive drug delivery to a specific location or collection of the microcontaminant from the environment with greater efficiency. We also expect that this swarm robot will be useful for the micro device that can detect pathogen or genetic information, or as a microreactor by assembling nanoparts together in an efficient way. Here I'd like to acknowledge all the contributors to this project. If you have any query related to this work, please send me an email. I'll try my best to support you. Thank you.